through this. So the first thing, and very important, that I have noted in your definitions is to know what a saccharide is. Firstly, so we're talking about hydrated carbons, which are basically carbon, hydrogen, and also oxygen. And these can vary in a number of ways, as you can see. So we have monosaccharides, disaccharides, oligosaccharides, and also polysaccharides. And of great note is monosaccharides, which, although there are many biological monosaccharides, the most important ones that we'll discuss are glucose, fructose, and galactose. That's because these are found in our foods primarily. And they can be interchanged into other things, of course. So disaccharides are, of course, made from our monosaccharides, which can vary, even up to maltose, which you may not be as familiar with. But there's also one called trehalose as well, which is also glucose and glucose, but it's bound a bit differently in terms of its structure. That goes into oligosaccharides, and lastly, polys, which are very important when we discuss starches and glycogen, as well as dietary fiber. So please know these different types, know the what they're made of, and that sort of thing. All right. So as far as monosaccharides, these are your single sugars. Saccharide, sugar, same thing. Glucose is considered to be probably the most important and prominent monosaccharide in terms of nutrient metabolism, as it is most widely used and also produced and preferred by various tissues of the body. And for that reason, we call glucose blood sugar, so a preferred source of energy. So it's found in fruits, vegetables, as well as honey. Next is fructose, which is found in fruits, honey, corn syrup. This is our fruit sugar and is very sweet. Of note is that fructose is chemically the same but has a different structure, as it looks like a pentose. So even though it has 6 carbons, same number of hydrogen and also oxygen, it is to that of glucose and galactose, it is still structurally different. And we'll look at that. So as you can see here to the right, it is not the same. It looks like a pentose sugar. However, it is not. Different to glucose and galactose. And lastly, galactose is, as far as I'm aware, is only bound to other sugars, or and not alone. Okay, so it's found bound to other sugars and not normally alone. So it's also important to note that galactose should not be confused with lactose as a disaccharide. In fact, lactose is made of galactose and glucose. And that brings us to our disaccharides. And these are our 2 linked sugar units. Of course, sucrose is glucose and fructose, our table sugar. Lactose is glucose and galactose. Maltose, of course, glucose and glucose. And trehalose is also glucose and glucose. However, its structure is different to that of maltose, which I'll show you in the next slide. So of note is, so in order to digest these disaccharides, what do we use? We use what's called a disaccharidase. Okay, a disaccharidase. So these are digest in the brush border, found in the glycocalyx, the unstirred layer of the brush border of the small intestine. Secreted or produced and secreted by enterocytes, we find our disaccharadases to digest these 2 sugar molecules. And these are specific to the different sugars, because we have sucrase for sucrose, we have lactase for lactose, we have maltase for maltose, and we even have trehalase for trehalose. Pretty cool. So we can't digest disaccharides in the mouth or the stomach. This occurs in the small intestine. So also you should refer back to table 2.1, and you'll see the various enzymes that are produced by the small intestine. You'll note that you'll see that these are produced in the small intestine. Another key thing here is in terms of the different disaccharides, maltose is of particular importance because it is produced in multiple pathways or ways in a common intermediate product of carbohydrate digestion. So when we have a starch, we also end up producing maltose as a result. We'll come back to that though. Okay, and another key important thing is how do we produce disaccharides? These are produced from a process known as condensation, or it's also known as dehydration synthesis. We call it this because in the formation of a two-sugar molecule, we form water and we then remove it. Okay, so water is formed from the combination of two hydroxyl groups, as you can see here. These would be a hydroxyl group on this sugar of this glucose and one on this glucose. From this, we yield H2O. And structurally, the hexose monosaccharides are very similar, with the exception of fructose. So as you can see here, fructose is again looks like a pentose sugar. However, technically is not because it contains 6 carbons. Hydroxyl groups are positioned differently depending on the carbon in all of these different monosaccharides. And most of these disaccharides that we digest are bound by alpha glycosidic bonds. Okay, glycosidic bond just means a sugar bond. So they're bound by glycosidic bonds. Again, these are alpha, meaning that this hydroxyl group goes upward. So this is beta. So beta, the alpha carbon, the hydroxyl group is directed upwards, whereas the alpha is directed downwards in space. So spatially, this one goes downward. Spatially, in the beta carbon, this one goes upward. So that bond would form a beta carbon in lactose, beta 1,4, and this would be alpha 1,4. Okay, the same thing for sucrose as well. I think that's it for this. So again, also to note is the fact that with things like, for instance, maltose, what does this 1,4 mean? So if you look here, these are both glucose, the same structure. However, this is the first carbon of the first or second glucose. So 1, 2, 3, 4. And on this glucose, we have 1, 2, 3, 4th carbon. They're bound, they form a bond, a glycosidic bond, at the first on this one and the 4th carbon on this one. So it's a 1,4 linkage. This is important when we come back to talking about enzymes that digest these disaccharides. If you note here, the trehalose is a 1-to-1. That's because it's on the first and the first carbon of both of these glucose molecules. Pretty cool. Okay. And another thing to discuss, it's not that important for this class. You've heard this stuff in OChem, but in terms of monosaccharides, there is a level of a stereoisomerism, which is just the occurrence of a molecule in different spatial configurations. So in space, they have some different type of how they're arranged in space. For instance, if we look at glyceraldehyde, actually go back to this. So this brings us to the chiral carbon. So it's based on the chiral carbon, which is the carbon in which we have 4 different atoms or groups that are attached to it. So here we have glyceraldehyde for instance. Why is this thing not moving? Okay, here we go. So this carbon here, the second carbon on a 3-carbon molecule, we have one group, we have the second group, which is just a hydrogen, and we have it's like an aldehyde here, and our last group here. So that's 4 different groups bound to this single carbon. And in terms of these epimers, what they call epimers or monosaccharides, this hydroxyl group is what dictates the level of or how it's. Let's see how. Let's see this. To go back, okay, so this is an enantiomer, okay? All these has an enantiomer, all right? Meaning that they can be mirror images of one another. So we have D-glyceraldehyde and L-glyceraldehyde. So D meaning right-handedness and L meaning left-handedness. So the hydroxyl group is spatially arranged on the right side of this chirocarbon, whereas the L-handedness one is on the left side. Okay, so it makes sense. In terms of metabolism and physiology, biology, or I should just say metabolism, we only deal with our enzymes only interact with D enantiomers of glucose, these monosaccharides. Okay? Only deal with the D. Yes, our enzymes only react with D isomers of these various sugars. That's important to note. That's really it. Alright, and here we have the, there we go. So all of these are considered to be either an aldehyde or aldose, or a ketone or a ketose. So for instance, glucose would be considered an aldehyde, fructose would be considered as a ketone. And this is based on the anomeric carbon location. So here on glucose we have an anomeric carbon, which is the carbonyl group here. So wherever you find that carbonyl group, that functional carbonyl group, you have your anomeric carbon. You see here that this is an aldehyde group, so it's an aldose. Here we have fructose. Where's the carbonyl group? It's on the second carbon. So this is considered to be a ketone group, or ketose. We'll talk more about this with glycolysis, though. Not so much today. All right. And as far as our oligosaccharides, so these are 3 to 10 saccharide or sugar molecules bound together, and these can vary quite a bit. Our most important ones, though, are raffinose, stachyose, should add this saved, verbiscose, and also maltose. There it is, okay. But to go back to the three previous ones, raffinose, stachyose, and verbiscose, you'll note that these have pretty much the same overall composition, but they vary, you could say, in terms of how much. Glucose, fructose, and galactose, but they all have galactose. That's the key thing here. And for that reason, you would need some type of galactosidase to digest these different oligosaccharides. However, we don't produce the enzymes to do it unless it's lactose, which is a beta-galactosidase. We don't produce the alpha-galactosidases, however. However, you can use BINO to give you the enzyme to digest these. Maybe, say for instance, you had a date, and I've done this so many times. You have a little pill, get your alpha-galactosidase, maybe get some, I don't know, some burritos or something like that, and you're good to go. No gas. That's because the bacteria in your intestine, they break down the, they digest the oligosaccharides, and a byproduct is, of course, the gas. All right. And lastly, at the bottom here, we have maltose, which is produced by, it can be actually industrially produced, but it's a result of starch digestion. So if I had a starch, one product of that would be maltose, which is an oligosaccharide string of straight 1,4 linkages of glucose monomers. Okay, so again, it's just glucose. There's no galactose or anything in it, but it is considered to be an oligosaccharide. maltose. All right, and so just to sum this up right here, our complex carbohydrates, these are oligo and polysaccharides. So please know each of these as we have discussed. So if I ask you about the digestibility of the oligosaccharides, raffinose, stachyose, and riboscose, what would you tell me? Think about it. Come back to it. And now let's talk about starches and glycogen. But there's also cellulose and gums or fiber that are also polysaccharides as well. This is not really important, but it's just kind of cool to see that plants have a very similar structure to that of muscle. Thought I had I don't have it. Okay, I don't. But if you look at a skeletal muscle fiber or muscle fiber, you'll see that it has similar, has fibrils. So it's made of amino acids, which make up into proteins called like actin and myosin, which make up filaments, which can make up muscle fibers, which can make up a large muscle. Same thing here with a plant, with a stem. It goes from very small molecules up to a macromolecule or large structure, organ. Pretty cool. So it's all about those fibrils. Okay. Now, I like to talk about it is glycogen. Pretty cool topic. That's because we produce it in the body. So animals and humans, or if you think of humans and animals, that's fine too. We produce carbohydrate. Well, we can of course produce carbohydrate, glucose, and we also can store it. This storage form is called glycogen. Glycogen is pretty important because, again, it is our storage form of glucose in the body, and it has some very important roles. For instance, with the liver, which stores not nearly as much as skeletal muscle does, only about up to 100 grams, whereas skeletal muscle can be up to 900 grams. The liver is what's called altruistic because it donates or gives glucose to all parts of the body, whereas skeletal muscle is very much selfish. This is because, well, we'll talk more about that later in a later day, but I won't talk about it now. Anyway, let's see. So glycogen, very important for exercise. Any type of high intensity workout or movement, you would need glycogen. Or not you need it, but it would definitely help. Or if you're starving, you would need, or fasting, you would need glycogen storage. All right. So the structure is very similar to that of amylopectin, which is a type of starch. Okay. Both of these are branched sugar molecules, and I'll talk more about why they or what that branching means in a second. What you should know is one that they're found in, or it's produced in the liver and also skeletal muscle. It's produced in all kinds of muscle, but predominantly in skeletal muscle. It's our major source of carbohydrate energy for active muscle during exercise, and also for maintaining bodily glucose levels. Don't judge me. A key thing to note also is the processes. So one process, in order to degrade or break down glycogen, it's called glycogenolysis. If you look at the name, lysis to split, and then just glycogen. So process O of splitting glycogen. And also it's called glycogenesis. This is to synthesize glycogen, and that's done in both of those places. Now, in terms of the branching, this occurs at the first and the sixth carbons on each of these two molecules, or these types of sugars. And when I say that, I'm sorry, we're talking about amylopectin and glycogen. Alright, so if you look here, there's an alpha 1,4 bond. So those are normal glucose to glucose, like maltose or maltose, which would be, or dextrin. Oops, sorry. However, from that we can produce a branch, and that will be from a 1 to 6 linkage. Okay, please make sure you know that. Both glycogen and amylopectin have these branches. And so these 1 to 6 bonds, they require a different enzyme for digestion or degradation to release glucose. Which brings us to this enzyme. This slide. Okay, so in our discussion about starches and polysaccharides, very important to note are the enzymes themselves which digest them. So we have our amylases, so salivary and pancreatic amylase. These are alpha 1,4 amylases. Excuse me. So 1,4 meaning, as you already know, the linkages, the glycosidic bonds that occur. So again, the fourth carbon on one glucose and the first carbon on the next. So here we have amylose, which is a string of carbons. String of glucose, straight chain. Okay, and we have our amylopectin, which is both straight chain 1,4s, and also we have our 1,6 glycosidic bonds. So on the sixth carbon is bound to the first carbon on another glucose to form a branch. All right, so make sure you understand that. Make sure you know that. Now, this occurs in both the mouth, so salivary, as well as the small intestine, produced by the enterocytes, the pancreatic, not the enterocyte. I'm sorry, the pancreas. The pancreas produces the amylase. All right. Now, in order to digest, it's called a dextrin. So dextrin is just a product of enzymatic hydrolysis of starch. We end up getting called dextrins, and these can vary. A very common one you may have heard of, maybe in sports supplements or food, it's called maltodextrin, which is a polysaccharide. But we also have what's called limit dextrins, and these can be oligosaccharides, which have a 1,6 linkage or branch. Okay. For this to occur, for digesting these, we need what's called dextrinase, or it's called isomaltase. Actually, this enzyme would digest both 1,4 linkages as well as 1,6s, but of most interest is its digestion of 1,6 linkages of limit dextrins. Okay. So as a result of starch digestion, we get maltose, which is just 3 glucoses, and we also get limit dextrins and other dextrins. For digesting these branches on those dextrins and limit dextrins, you need dextrinase. In addition, we have what's called resistant starches, which are ultimately resistant to these amylases and other enzymes, or resistant to alpha amylase 1,4. We'll talk more about that in the fiber section. Very important to note also is the fact that in the stomach there is very, very limited, if any, in fact, just to make this clear, carbohydrate digestion will cease in the stomach. And again, this is due to the amylase being denatured. The protein is actually unraveled and becomes inactive in the stomach. And this is a result of a very low pH. And once again, as I've already discussed, Those disaccharidases produced by the enterocyte in the brush border are secreted into the brush border, the glycocalyx. We have lactase, sucrase, maltase, and also our alpha dextrinase, as well as trehalase. These are enzymes which will utilize hydrolysis, so the use of water, to split these various disaccharides. And here, let's see, in order to absorb and, of course, transport these various monosaccharides after they have been digested, we have two primary processes. One is active transport, so with the use of ATP, and also a transport protein, sodium. We have the absorption of glucose as well as galactose. But we also have facilitated diffusion or facilitated transport, which is using just a transport protein. We have our, in terms of active transport, we have sodium glucose transport, or sodium dependent glucose. It's called symporter, meaning that both sodium and glucose are transported at the same time, or utilized by this protein at the same time, or absorbed into the cell. And this is fueled by a sodium-potassium ATPase, or sodium-potassium pump. Now, what happens here is a concentration gradient is created in which sodium is pumped out of the cell. Okay, pumped out of the could be into the bloodstream, could be pumped out of the cell, and this creates a gradient to increase the pumping in of sodium from the other side. The other side is called the apical membrane. The apical membrane is from the lumen of the small intestine to where the cell is. Okay, so the inside of the cell and the lumen of the small intestine, that membrane is called the apical membrane. Now, on the other side of that is called the basolateral membrane. This connects the enterocyte to the bloodstream, okay, to the peripheral blood, or it could also be a lymph vessel, for instance. All right. Now to continue, we have again, as I was saying, the facilitated diffusion. With that said, we have something called GLUT, a GLUT transporter, or glucose transporter 2. Now this is the same for all three monosaccharides. This allows for transport of that monosaccharide from the enterocyte into the bloodstream, called GLUT2, which we'll talk more about in a second. Of note also is the fact that SGLT1 is utilized by both glucose and galactose, but not fructose, apparently. And also during high concentration carbohydrate meals, we have increased use of GLUT2 during that time. In fact, looking at this study here, the result of the study, if you delete the SGLT1 gene in mice, this results in glucose accumulation in the gut. Pretty cool. You can see here that those dark dots, these represent mice that would have the SGLT1 gene, so they're still producing the protein, okay, the transporter, whereas these open squares would not have that gene, and thus they accumulate more and more glucose where they shouldn't, so it stays in the intestine. Pretty interesting. And this shows us our various, at least some of them, transport proteins. So again, SGLT1, active transport, allowing for sodium and glucose to symport, okay, into the enterocyte. And on the other end of this, we have GLUT2, glucose transporter 2, to allow for glucose, galactose, and fructose to leave the cell and cross the basolateral membrane and into the bloodstream. Now for fructose, we have something different, and that is, it's called GLUT5. GLUT5 is used supposedly exclusively for fructose. Okay, so the other two do not utilize this particular transport protein. Also of note is the sodium potassium ATPase here, which created that concentration gradient to allow for the pumping in of sodium with glucose into the cell. In addition, lastly, again, high intake of, I didn't say this, GLUT2 can also fuse to the apical membrane here of the brush border to absorb glucose and galactose. Okay, that same transport protein can be used on the other side of the basolateral membrane. Okay, GLUT2 can be used on both sides of the fence, and this is important during higher intake, high carb intake meals. So I've already kind of mentioned this, but once again, so absorption of fructose into the blood, GLUT2, but from the lumen of the cell, from the apical membrane, we have GLUT5. Please don't forget that. This is somewhat limited in about 60% of adults, and that you would have more fructose being left in that area for longer. This creates some type of stomach or some type of gastrointestinal distress. And typically there's no fructose beyond the portal vein, as it is efficiently absorbed by the liver. It is, however, absorbed slower than glucose and galactose, but faster than sugar alcohols. So yeah, so fructose often causes GI distress with high intake. So for instance, one thing I always talk about is, if you eat a lot of fruit before you go for a run, this is a bad idea because this will most likely cause you some GI distress and limit your ability while you're trying to run or do some type of activity. Okay. And once again, facilitative diffusion or transport, we've already talked about that, so we're good to go. Well, I'll say more about it, but in terms of the glucose transporters, these are very special. Um, they're highly polar, as glucose is highly polar. Um, Transport system to get glucose in and out of cells, um, various cells, and all kinds of cells in the body, not just the enterocyte, but all cells in the body actually. And there are about 14 of these GLUT transporters, glucose transporters, called GLUTs. Not your hamstrings, but similar. The name. All right, so there's 14 identified with genes. They are um, somewhat integral proteins, or they become integral into the lipid bilayer. They will embed themselves in the lipid bilayer from within the cell, from vesicles, lodge themselves into the plasma membrane, and then go back as a process known as endocytosis. So the endocytose and form a vesicle and go back into or leave the membrane. Pretty cool. Okay, so here it says that insulin at high levels promotes GLUT2 anti-translocation back to secretory vesicles. Uh, so this state represents a high blood glucose level. So if you have high insulin, this means that there's a high blood glucose. So it makes sense because insulin, what does it do? It stimulates glucose uptake into cells. In this instance, question is, how does this affect the GI tract or the enterocyte? Well, it's going to prevent more glucose from being absorbed at that time. So thus, it's a way of controlling the rate of more glucose entering the bloodstream. So it makes sense. So recall that GLUT2 is used for exiting the enterocyte and entering the portal blood. So insulin can actually prevent excess amounts of glucose entering the bloodstream. So here we have some pretty important ones. GLUTs 1 through 5. Of more note, though, is definitely is going to be GLUT4, which is insulin dependent, which we do find predominantly in muscle, fat cells, adipocytes, as well as the heart as well. But you see others. There's many different cells in the body which utilize these GLUTs to get glucose. Yeah, so fructose for GLUT5, but not glucose. So insulin, it initiates the activity of GLUT4. So I think everyone knows this, but insulin is critical for, or not critical, but it's very important for glucose uptake and absorption in cells, various cells in the body. But as you can see, is this the only way that glucose gets into the cell? Is it just through insulin? No, it isn't. There are 14 other, GLUTs. That's how important glucose is. In terms of how this happens, we had GLUT4 movement to the cell membrane. As you can see here, so coming from the trans Golgi network, okay, which is connected to the endoplasmic reticulum. This Golgi body will bud, the budding process, form a vesicle, and it is here in which these transport proteins are stored within the cell. Now, upon stimulation by insulin will bind to its receptor, as shown here with the insulin receptor. This will stimulate the lodging of GLUT4 to the cell membrane, okay, tethering, docking to it, in which it then fuses through a process known as exocytosis, as shown here. And these proteins will then allow for glucose to be uptaken, absorbed into the cell. Hope that makes sense. Now, when it's done, or when insulin has stopped stimulating the cell, it will then endocytose and then go back to where it came and form a vesicle until the next time there is more stimulation. And that's the process. Here it's shown once again. But to add to that, pretty cool to me, is the fact that not only can insulin do this, but also through AMPK, adenosine monophosphate kinase. This is how the molecular process in which it is transduced. We have the translocation of GLUT4 to the membrane for glucose uptake as a result of muscle contractions. That's pretty cool, because just through exercise, we can actually stimulate and more so, I would say, sensitize cells to glucose. Or insulin, I would say, to glucose. Sensitize those cells for proper uptake of glucose into the cell. So a big problem for us today is the fact that we're sedentary, and we have lack of this occurring. So So just by being more active, you get better glucose uptake through these GLUT4 transporters. Isn't that awesome? And even here you can see with this study by Funaki in 2004, with no treatment of insulin, we see here this green staining represents GLUT4 proteins. Now, upon stimulation by insulin, they will then leave within the cell, or not within the cell, but you could say, I guess, the middle of the cell, and lodge into the plasma membrane. Once it's gone, it goes back to this. It's awesome. In terms of the glycemic response to carbohydrates, we have something called the glycemic index and glycemic load, which you guys are probably familiar with already. It really isn't that important unless you're very concerned about maybe you're diabetic and you only consume one type of food at a time. You know, like if you want to know about candy, what kind of effect this has on your blood glucose, your blood sugar. Of course, what's going to happen is it will spike insulin as well as glucose at that time. Whereas if you consume something which would have more fiber, which has a slower release of glucose into the bloodstream, this would, of course, have a lower glycemic index and thus most likely lower load, which is based on the quantity or gram per gram, as well as the glycemic index. Really not that important, honestly. And this is all compared to a reference food, which could be, you know, white bread, or it could be some white rice, something that would have a very strong effect on blood sugar. And this table shows us some various responses that we would see, or glycemic indexes, as well as loads from various foods. I think this is our last slide. I would like to make a correction that you might see in your slides, which I don't know how it happened, but it was wrong. Glycogenesis is the process of synthesizing glycogen. It is not the process of synthesizing glucose. Okay, please make sure that's correct. Glycogenolysis is the degradation of glycogen, which does occur in both the liver and as well as the muscle. Glycolysis, this is the oxidation of glucose, or the degradation of glucose, to produce eventually carbon dioxide and, well, actually, let's be specific here. Glycolysis produces ATP and NADH, right? Okay, we also have gluconeogenesis. Now, this is the process in which we synthesize glucose, and this is, you could say, de novo synthesis, so fresh synthesis, new glucose from non-carbohydrate intermediates. So it could come from amino acids, from even a fat-derived molecule called glycerol. It could be lactate. That's gluconeogenesis. We'll talk more about that in 434. There's also pentose phosphate pathway, or hexose monophosphate shunt. So we'll say PPP or HMP shunt. This is the synthesis of 5 carbon, or so pentoses, monosaccharides, and also it's called nicotinamide adenine dinucleotide phosphate, so NADPH, which is a very, very important coenzyme reducing agent. We'll talk more about that later. Lastly, we have the tricarboxylic acid cycle. It's also known as the Krebs cycle and also the citric acid cycle. So this is the ultimate, or at least one of the ultimate, the oxidation of pyruvate and acetyl-CoA to produce CO2, carbon dioxide, as well as eventually water through the electron transport chain and oxidative phosphorylation.